Hello again. Um, this is Biosignaling 1.2. It's a complementary lecture to the previous one, Biosignaling 1.1. In Biosignaling 1.1, I explained some basics of biosignaling and cell signaling in general. I defined biosignaling, biosignals. I talked about different types of ligands, different types of messengers, and so on, and also receptors. In this complementary lecture, I'm going to talk about a little bit about the quantification of receptor-ligand interactions. Then I'm going to talk about some common features of signaling pathways. I'm going to talk about two of the most important types of responses. Different types of responses. In the previous lecture, I told you that there are different responses. For example, secretion or movement, or mitosis or apoptosis. But now I'm going to talk about another classification of responses, some other classification. Sorry about the outside noises. Intracellular signaling complexes is the last thing that I'm going to talk about in this lecture. I'm going to define them. Okay. Oh, didn't see that coming. Receptor desensitization also. It's another thing. Types of receptor desensitization, and how a receptor is desensitized or is adapted to a signal. Okay, let's begin by quantification of receptor-ligand interactions. As we talked about it in the previous lecture, like a ligand, for example, it can be a hormone or any other a signal molecule. This ligand or hormone binds tightly and specifically to the receptor R. These two words are very key words. First of all, the bond is tight. It's not as tight as covalent bonds, of course, but it's tight and it's a specific. There is a molecular specificity between the receptor and its ligand. Okay. The bond we are talking about here, the bond between the ligand and the receptor, is mostly mediated by non-covalent bonds. Hydrophobic interactions, hydrogen bonds, electrostatic interactions or ionic bonds, van der Waals interactions. So, these are all examples of non-covalent bonds, and they play a role in receptor and ligand binding. The hydrogen bonds, the electrostatic bonds work both in short and long distances, and they're both repulsive and attractive. But van der Waals interactions only work within short distances, and they're always attractive. Okay, most of them work for small ligands. Most of these bonds work for small ligands. For large ligands, as we saw, for example, an epidermal growth factor as an example of a very large ligand, hydrophobic interactions may also play a role. Okay, we have it here. There's an exception. There are some exceptions. I guess, no, I didn't understand that part of a text that I studied for this, but I think that there's a signal molecule which is covalently bonded to rhodopsin or light-sensitive receptor in our retinoids. So that can be an exception to these non-covalent interactions. Okay, that's maybe the only one which is mediated by covalent bonds or interactions. But most of them, because we need the ligand to be temporarily bounded to the receptor. We want it to be reversible. That's why most of the bonds are mediated with non-covalent interactions. So what happens when a ligand is attached to a receptor? There's going to be a conformational change. It means that the shape or the 3D structure of a receptor undergoes a change, and that conformational change, structure determines function. When the structure of a receptor changes, its bioactivity or its function also changes, and that's how a ligand somehow activates a receptor by changing its conformation, thus and in turn changing the bioactivity or the function of the receptor. In the case of, for example, a G protein coupled receptor, which is attached to a protein, a G protein, the G protein coupled receptor, when a ligand binds to it, it undergoes a conformational change, and that conformational change somehow activates the G protein, and the G protein acts as a transducer and goes on and affects some effector proteins. Okay, we're going to have a lot of those examples of those conformational changes in other lectures, in future lectures. There's a very simple equation for receptor and ligand binding. So we have R plus L gives us the RL, which is the receptor-ligand complex. This two-headed arrows, this symbol represents, in chemistry, represents, a chemical reaction which can happen in both directions. In here, we mean the reversibility of receptor and ligand interactions. It means that this receptor-ligand complex is not permanent, and the ligand is going to detach itself from the receptor at some point, okay, after it delivers the message. I got this the information in these slides from the Principles of Biochemistry, Lehninger's Principles of Biochemistry. Yeah, Principles of Biochemistry. Okay, so the receptor-ligand interactions or binding depends on the concentration of interacting parts. And that's why it's saturable, okay? The receptor binding, ligand binding is saturable. I mean, if, we have a limited number of receptors, okay? And when they are all occupied, when they are all attached to a ligand, okay? If you add more ligand to the environment, they're not going to do anything because we don't have enough receptors. So, so it's very important. The equilibrium constant, I'm not going through all this, chemical formulas, but generally, the concept that I want to introduce here is this K of A, or association constant, which is equal to the concentration of those receptor-ligand complexes divided by the concentration of individual receptors or unoccupied receptors and individual ligands. And it is the opposite or reverse of the dissociation constant. So Ka represents dissociation constant, and dissociation constant represented by Kd. Basically, these two constants are the measures of the affinity of a receptor for its ligand. So, for example, the lower the dissociation constant, the higher the affinity of a receptor for its ligand. For example, for the dissociation constant for some hormones is for example 10 to the minus 9 or 10 to the minus 11. So it's very low. That means that hormones or have very high affinity for their receptors. Okay, that was it. That was all I wanted to talk about, the quantification of receptor-ligand interactions. Oh, features of signal transduction pathways. One of the sorry. One of the basic features of any signal transduction pathway is the specificity between the signal molecule and its receptor, between the ligand and receptor. This is a result of the molecular complementarity between the ligand and the receptor. One of the ways, if you remember in the previous lecture I told you that a cell may be bombarded with different and various kinds of signals. But one of the ways that cells can identify a very specific signal molecule or a very specific ligand is because each ligand has a specific receptor, okay? And it can only attach to that. There are some exceptions, but most of the ligands only attach to their specific receptors. And so specificity is a very important and key feature of almost all of those signal transduction pathways. Another important feature is the amplification. Again, in the previous lecture, I told you that there are second messengers as well as first messengers. First messengers are extracellular signaling proteins. Second messengers are intercellular or intracellular signaling proteins. And what they do usually, is that they amplify the signal inside the cell, or they amplify the signal carried by the first messengers. But sometimes transducer proteins can amplify the message by the first messengers. For example, in this case we have a signal, okay? And that signal, or our first messenger here, activates enzyme 1. Enzyme 1 can be a transducer. It only needs to amplify one, activate one of those enzyme 1s. Okay? Each one of these enzyme 1s, going to activate a number of enzyme 2 types, enzyme 2s. Each one of these enzymes are going to activate a number of other enzymes. This is how a signal is amplified. So one signal is enough for activating one type of enzyme. That type of enzyme are going to activate a number of other types of enzymes. Each one of those enzymes are going to activate a number of other enzymes. And that's how a single signal is amplified and is spread throughout the cell. Amplification is a very important feature to spread the signal. It also increases the efficiency of first messengers as well. Modularity. There are some scaffold proteins. For example, here you see when the signal comes in, you see that we have this signal protein, this transducer, we have this transducer, and we have this transducer. They're all attached together in small modules. And look at this, it is like a scaffold for this one. The last one is like a scaffold for the second one. There are some scaffold proteins which pre-assemble intracellular signaling proteins to increase the efficiency of the interactions between them. I'm going to explain different types of intracellular complexes. So, just have this for modularity. Desensitization or adaptation of a receptor is also another feature. When the signal is present in the environment for a long time, the receptor is going to be desensitized it's going to be adapted to it. It's going to adapt itself to it. Since we don't want a signal to be always present, okay? So if the signal is constantly present, the receptor is going to be desensitized and no longer is able to, get the signal. Again, I'm going to talk about different types of mechanisms of receptor desensitization. The last common feature of signal transduction pathways is the integration of 2 or 3 or more signals. Again, I told you that a single cell may receive various types of signals. Obviously, those and each one of those signals are going to initiate their own signal transduction pathways in the cell. And those signal transduction pathways can sometimes interact with each other. But for example, look at this example. There's signal 1 received by receptor 1. Its effect is to, for example, increase the concentration of substance X or increase the membrane voltage or membrane potential. The signal 2 is received by receptor 2. At the same time, it has completely opposite effects. For example, it decreases the concentration of substance X or it decreases the membrane potential. But the response is going to be based on the net result of these two signals, or net result of these two signal transduction pathways. And one of the challenges of the modern cell biology and modern system biology is to understand how cells make sense of all of these complicated signal transduction pathways and how they respond based on the integrated or the net change that these different signals cause. And so it was about integration of various signals. Okay. There are two general types of responses. There's a slow one and a rapid one. Let's see what happens in each one of them and why we call a certain type of response slow and why we call a certain type of response rapid. So we have an extracellular signaling molecule received by this cell surface receptor protein. Okay. So this is an intracellular signaling pathway, those molecular interactions between transducers and effectors. We're going to have altered protein function. How protein's function is altered? There are some transducers, there are some enzymes, for example, like kinase enzymes, which add a phosphate group to a protein. The addition of when they add a phosphate group to a protein, they make that protein active, okay, or sometimes deactive. And that's how a protein's function is altered. And that altered protein is going to alter some cytoplasmic machinery or protein machinery, and at the end, this leads to the altered cell behavior, or in another term, this leads to a cell's response to that very first signal we had here. This alteration of a protein function is temporarily, and it can happen very fast thanks to those enzymes that I told you. For example, for instance, kinase enzymes that they add a phosphate group to other proteins or other molecules to make them active or inactive. And it's not permanent. Okay, it's temporarily. That phosphate can be easily removed by another enzyme, and it will. And it only lasts seconds to minutes. And it happens very fast. Again, I told you, because of those enzymes. So this is why this is called rapid response, because the events which are required for altering cells' activity happen very fast. And since the events are not permanent, or they're not going to last for a long time, they're also called short-term changes. So the alteration in protein function is a short-term change. Okay? Let's see, what do we mean by a slow response? So again, imagine we have another extracellular signaling protein received by a receptor. But here, it activates some transducers or transduction proteins. They get into the nucleus. They should activate some transcription factors. So first they have to get into the nucleus. They need to activate some transcription factors. They need to recruit some transcription machinery to the DNA. They need to synthesize RNA. The RNA should get out of the nucleus. And here we have altered protein synthesis. And the protein synthesis itself takes time. And then we have altered cytoplasmic machinery. And then we have the cell's response. You see the number of steps required for in this pathway outnumbers the number of steps in this pathway. That's why it is slow. It takes minutes to hours, okay? Because as I explained, it should, different molecules should be translocated and should be transported into the nucleus, and then they should recruit other proteins. New proteins must be synthesized, and that's why it takes much more time than this pathway. And also because new proteins are synthesized, they're going to stay in the cytoplasm for relatively longer times. So we can call these changes long-term changes because new proteins are synthesized. There's another way to call these, to name these two changes. Short-term changes are usually functional changes, okay? As we had here, there's an altered protein function, so there's a functional change. That's why it's short-term, and it also happens very fast. But long-term changes are also called structural changes. Why structural? Because we have synthesized new proteins. That's why they're called long-term changes or structural changes, and that's why they take much longer time to occur. And a spoiler alert, this is the basis of short-term and long-term memory. Okay? I'm going to explain in implicit and explicit memory lectures, but this is actually the basics of that. The basis of that, sorry. Okay, now intracellular signaling complexes. As I told you in the part of the modularity, when I was talking about the modularity as an important feature of different signal transduction pathways, there are some proteins which pre-assemble transducers and transduction proteins, transducing proteins, or intracellular signaling proteins. Let me show you the example, actually. So, for example, here, we have an inactive receptor. We have a scaffold protein. What it does is to pre-assemble some intracellular signaling proteins, okay? These signaling proteins, intracellular signaling proteins or in transducers, should be activated in sequence. Instead of being in different locations in the cytoplasm, they're in one single place. They're held and pre-assembled in one single place by a scaffold protein. So when a signal molecule comes in, received by the receptor, they're activated by the receptor in sequence, and they go to downstream signals and they do other stuff, okay? But they activate in sequence. This is very important because it increases the efficiency of signaling. It takes less time to activate all of them in sequence. Another important role it has is that it prevents unnecessary interactions between these signaling proteins. Signaling complexes actually is one of those mechanisms that cells have came up with to prevent crosstalks between different signaling pathways. Yeah. We have another type of signaling complex. For example, in this case, we don't have the scaffold protein. What we have is an inactive receptor. When the receptor receives the signal molecule, it gets activated. When it gets activated, it phosphorylates itself. It adds a phosphate group to itself, and also it phosphorylates other signaling proteins or transducers, and again they go and work in downstream signals. Okay. So you can see, yeah, right now in this type of signaling complex, the signaling proteins or transducers are not pre-assembled, but they can dock into these specific places and get activated. Okay. The last one is, again we have, an inactive receptor. Here we have a specific phospholipid called phosphoinositides. These phospholipids or phosphoinositides, when a receptor is activated, it phosphorylates these phosphoinositides, and they get hyperphosphorylated. Okay, they get overphosphorylated. When they get overphosphorylated, they act as a docking site for some of those intracellular signaling proteins. Okay? So, again, the receptor is activated. It activates or phosphorylates those phosphoinositides. When they're hyperphosphorylated or overphosphorylated, they act as a docking site for those intracellular proteins, signal proteins, and they activate each other in sequence, and again they go and work in downstream signals. This is how intracellular signaling complexes work. Now let's talk about desensitization to an external signal molecule. There are various ways and various mechanisms for to make a receptor desensitized to an extracellular signal molecule. The first one is temporary sequestration of the receptor. So when a signal molecule is attached by the receptor, it is detached from the cell membrane, gets into the endosome, okay, and the signal is released, and then it is, temporarily sequestrated in the endosome, and after a short time or some time, it gets out when the endosome actually fuses with the cell membrane again. This receptor protein recycled and gets back to the is recycled and gets back to the plasma membrane. So this is one way. The next way is that instead of getting into an endosome, the receptor gets into the lysosome. It's destroyed. So-called downregulation of receptor. Okay, it's destroyed, and so it's no longer available for the signal. Sometimes the receptor is inactivated instead of being sequestrated or destroyed. Sometimes it's inactivated. The way a receptor gets inactivated is that, for example, there are some proteins which add an extra or a phosphate group to a receptor. That phosphate group, for example, prevents the receptor to go under conformational change, so it's no longer able to receive a signal. Okay, that's how, for example, the receptor is inactivated. Phosphorylation or dephosphorylation sometimes can inactivate a receptor. Sometimes the signaling protein, which is related to this to the receptor, gets inactivated. For example, In a G protein coupled receptor case, we have, the G protein coupled receptor is attached to a G protein. When it gets activated by a signal, after binding to a signal, it activates a G protein. G protein, goes under some conformational changes. It takes time for the G protein to get back, okay, to the G protein coupled receptor. In that period, when the G protein is detached, the G protein coupled receptor is actually, we can say that it is desensitized to the signal, to a new signal. And sometimes there are some inhibitory proteins which are going to inhibit the, again, that attached transducer or signaling, intracellular signaling protein to the receptor, and by doing this, the receptor is going to be desensitized. So there are 5 different ways that a signal, that a receptor can be desensitized to a signal. Desensitization is very important because, as I told you, we don't want a signal to be always present or to be always detected by the cell. Okay? So that was it. It was a short complementary lecture to the Biosignaling 1.1. The next couple of lectures are going to be about the key molecules in common signaling pathways. I'm going to talk about G proteins, kinase enzymes, and phosphatase enzymes, how they work, their structures, and so on. And there's going to be another lecture, Biosignaling 3, which I'm going to talk about some other facts and principles and concepts related to biosignaling, and after that, explicit and implicit memory. Thank you so much for watching this video. I hope you can watch other videos too. Take care and have a nice day.